Every living thing, plant or animal, large or small, is composed of cells. Some organisms, such as the amoeba or paramecium, consist of only a single cell, while others, like human beings, consist of many trillions. What exactly is a cell? How big are they? And why are they necessary for life? You'll find the answers to these and other questions as we look at the cells, tissues, and skin of the human body. Although most are microscopic, cells vary widely in size. Human sperm cells are only about 2 micrometers in length. That's 1/12,000th of an inch. Some nerve cells, for example the ones that connect your spinal cord with your little toe, are longer than 3 feet. Cells also vary widely in function. Single-celled organisms like the amoeba or paramecium have the ability to carry out all the necessary life functions independently. For example, they can obtain and digest food, eliminate waste, and respond to a variety of stimuli. But in multicellular organisms, like human beings, cells don't operate independently. Instead, they've evolved highly specialized forms and functions that work together to allow the entire organism to survive. Although our cells differ in many respects, they all have certain characteristics in common. First, they all have a plasma membrane, the thin outer layer that separates the cell from its environment. Most also have a nucleus that contains their genetic material, called deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Cells also contain specialized structures called organelles and the semi-fluid cytoplasm that surrounds the organelles and nucleus. Let's take a closer look at each of these cell components and find out what they do. Cell membranes have the ability to let some particles pass through, but not others. Because of this characteristic, they are referred to as selectively permeable or semi-permeable. Their basic structure is a double or bilayer of phospholipid molecules. These molecules align themselves so that their water-soluble hydrophilic heads face toward the inner and outer surfaces of the cell, while the non-water-soluble tails line up in the center of the membrane. This way, the two layers lie tail to tail, exposing the heads to water inside the cell and out. Cholesterol molecules wedge themselves between the phospholipid tails, stabilizing the membrane structure. Transmembrane proteins span the width of the membrane to transport nutrients and waste products into and out of the cell. Peripheral proteins are attached to either the inner or outer surface of the membrane. The glycocalyx, a fuzzy coat on the cell's surface, provides highly specific biological markers that allow cells to recognize each other as self cells, that is, cells that are a part of your body, as opposed to cells like bacteria that are not. One of the most important physical processes to all living things is diffusion. But what is diffusion, and why is it so important?
Diffusion is the process by which particles spread from regions of higher concentration to areas of lower concentration until equilibrium is attained. This process is absolutely critical for cells to survive. For example, cells need oxygen to make energy. Because the cell is constantly using up its oxygen supply in its metabolic processes, oxygen is always in lower concentration inside the cell than outside. Because of this differential, the process of diffusion causes oxygen molecules to flow from an area of higher concentration outside of the cell across the semi-permeable cell membrane into the cell's interior, where oxygen concentration is lower. The same process works in reverse with carbon dioxide, a waste product of cytorespiration. In this case, the CO2 is in higher concentration inside the cell than outside.
Diffusion can distribute molecules rapidly over short distances, but it is extremely slow over distances greater than a few centimeters.
If the human body relied strictly on diffusion, it would take over 200 days for oxygen to travel from the lungs to the brain. Brain cells die in only 4 to 6 minutes without oxygen, so that's why multicellular organisms like ourselves cannot survive by diffusion alone.
Instead, our circulatory system with its oxygen-carrying red blood cells bridges the gap. It moves oxygen and other substances around the body much more quickly than diffusion. But at the cellular level, our bodies still rely on diffusion. In simple diffusion, substances such as oxygen and carbon dioxide are able to pass through the non-water soluble hydrophobic interior of the plasma membrane without assistance because they can dissolve in the lipid bilayer.
Their net direction of flow into or out of the cell depends on the concentration gradient, the difference in the concentration of like molecules inside and outside of the cell. Oxygen will always diffuse into a cell because the oxygen concentration is always greater in the blood than in the tissue cells. The opposite is true with carbon dioxide. So carbon dioxide will always flow from cells to the blood.
Larger particles of water-soluble substances, such as potassium ions, are unable to diffuse directly through the plasma membrane unassisted. But they can diffuse through channels within the transmembrane proteins. Most channels are primarily permeable to a specific ion, such as potassium. Lipid-insoluble substances, such as glucose and amino acids, that are too large to pass through the channel proteins may cross a cell membrane using a process known as facilitated diffusion. In this situation, certain carrier proteins appear to have a binding site for these larger molecules that is moved from one face of the membrane to the other. Like the channels of the transmembrane proteins, these binding sites appear to be highly selective as to what they transport. Another type of diffusion is called osmosis. The left side of this beaker is filled with a salt solution, while the right side is pure water. A semi-permeable membrane separates the two compartments. It allows water molecules to pass through, but not salt molecules. In a solution, solutes, in this case the dissolved salt particles, take up space that water molecules would otherwise occupy. This results in a lower concentration of water molecules on the solute side. Because of this, water will move by osmosis through the membrane from the compartment on the right with lower solute concentration to the one on the left with the higher concentration until equilibrium is reached. As a result, the volume of the right compartment decreases while the left compartment increases. So how does this process of osmosis affect a cell? There are 3 possible scenarios. In a hypertonic situation, a higher solute concentration exists outside of a cell than inside it. This exerts osmotic pressure for water molecules inside the cell to diffuse outside where the concentration of water molecules is less. This causes the cell to shrink due to net water loss. In a hypotonic situation, the exterior solute concentration is lower than it is inside a cell. Water would tend to diffuse into the cell, causing it to swell due to net water gain. Solutions that have the same solute concentration inside and outside a cell are considered isotonic. These solutions will neither cause the cell to take on water nor to lose it. In some situations, such as with blood pressure, water and solute particles are forced through membranes by hydrostatic pressure. This process is called filtration. Simple diffusion, facilitated diffusion, osmosis, and filtration are all passive transport processes, which means they do not require any energy to be expended on the part of the cell. There are also active transport processes that require the cell to expend energy. The first is solute pumping.
Solute pumping involves moving substances against concentration gradients using carrier proteins called pumps. This allows cells to keep their higher concentrations of different solutes inside their cell membranes than exist outside of the cell.
Let's consider the sodium-potassium pump found in nerve and muscle cells. This pump transports sodium ions out of the cell while moving potassium ions in. This keeps potassium levels inside the cell 10 to 20 times higher than those found in the environment outside the cell. This pump operates more or less continuously, but it takes an expenditure of energy to do this. Endocytosis is another type of active transport, where large particles outside the cell are engulfed or enclosed within small membranes called vesicles. The vesicle moves through the membrane and into the cytoplasm, where its cargo is released. Exocytosis is the same process in reverse and is often used to rid the cell of waste. Cytoplasm is a clear, colorless substance that fills the area inside the cell membrane and outside of the nucleus. It consists of three main elements: cytosol, the thick, semi-transparent fluid in which all other cytoplasmic elements are suspended; inclusions, chemical substances such as nutrients or cell products like fat droplets or glycogen granules; and organelles, the structures that carry out particular functions for the cell. The first of these structures is the endoplasmic reticulum, or ER.
ER is a complex organelle composed of membrane-bound, flattened sacs and elongated canals that twist through the cytoplasm, connecting and communicating with other organelles and the cell nucleus.
Rough ER has ribosomes attached to its surface, which synthesize all the proteins secreted from the cell. ER without the ribosomes is called smooth ER. Smooth ER manufactures certain lipid molecules rather than proteins. The Golgi apparatus is the processing and transportation center of the cell. It receives proteins from the rough endoplasmic reticulum, adds any finishing touches, packages them in vesicles, and ships them out to specific locations. Mitochondria are the cell's power plants. They supply it with adenosine triphosphate, or ATP, the energy needed to perform cell functions. Microfilaments are involved with cell movement and changes in cell shape. Intermediate filaments strengthen the cell and help resist pulling forces. Microtubules are long, hollow cylinders that help determine overall cell shape and the distribution of cellular organelles. Together, microfilaments, intermediate filaments, and microtubules form the cytoskeleton. Centrioles are a special type of microtubule important to cell division. The nucleus contains the genetic code to produce virtually every cell in the body. Nuclei are bounded by two lipid bilayer membranes. Nuclear pores regulate the entry and exit of large particles. The nuclear envelope encloses a gel-like fluid called the nucleoplasm, in which other nuclear elements are suspended. The nuclear lamina, a network of protein filaments similar to the cytoskeleton, lines the inner membrane and maintains the shape of the nucleus. Each nucleus contains one or more nucleoli, the sites where the subunits of ribosomes are assembled. The nucleus also contains long ribbon-like pieces of chromatin.
Chromatin is composed of equal amounts of DNA, our genetic material, and histone proteins. The histone proteins provide a physical means for packing the very large DNA molecules into the very small space within the nucleus of the cell.
Every cell in our bodies undergoes a series of changes called the cell cycle. The first stage is interphase, a period of growth and development when a cell carries out its normal functions. During the M, or mitotic phase, the cell undergoes 6 stages of mitosis: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. In prophase, chromatin containing the DNA is condensed into structures called chromosomes, which are joined in identical pairs at the centromere. Spindle fibers appear from the centrosomes, and the nuclear envelope and nucleoli disappear. In prometaphase, some of the spindle fibers attach to the chromosomes, causing them to move. In metaphase, the chromosomes that are now attached to the spindle fibers line up along the midline of a cell. Next, in anaphase, the spindle fibers pull sister chromatids to opposite poles of the cell. During telophase, the chromatin decondenses from chromosomes back to ribbons. A new nuclear envelope and nucleoli appear, and spindles vanish. Telophase is the end of nuclear division, but overlaps with cytokinesis, which pinches the cell in two. Completion of this cycle results in two daughter cells with identical sets of genes. Although daughter cells inherit the same set of genetic material, they don't all end up looking or functioning alike. Differentiation is the process whereby an individual cell expresses or turns on some genes and represses or turns off others. This causes each cell to grow and develop into a specific type with a specialized function. Groups of cells that work together for a common goal are called tissues. The human body has four basic types: epithelial, connective, muscle, and nervous tissue. Epithelial tissues cover body surfaces, line most internal cavities and organs, and are the major component of glands. Specialized epithelial cells in the saliva and sweat glands produce substances released outside the body. Other specialized epithelial cells produce hormones, such as adrenaline and endorphins, that are released inside the body. Connective tissues have numerous and varied functions. For example, blood is a fluid connective tissue that transports substances within the body. Bone is the most rigid connective tissue. It serves as a framework for the body and provides protection to our vital organs. Other connective tissues bind muscles to bone, fill spaces between cells, store fat, protect against infection, and help repair tissue damage. Muscle is the third basic tissue type. There are three different kinds: skeletal, smooth, and cardiac. Among their many functions, muscles move our bones, pump our blood, digest our food, and help us breathe. Nervous tissue, the fourth tissue type, makes up the brain, spinal cord, and peripheral nerves, which coordinate, regulate, and integrate many body functions. Tissues can also be classified by the shape of their cells and the number of cell layers they contain. Squamous cells are flat, cuboidal cells are cube-shaped, and columnar cells are shaped like a column or cylinder. Simple tissues have a single cell layer. An example would be the squamous epithelium that lines the air sacs in the lungs and the walls of blood capillaries. Stratified tissues have multiple layers of the same cell type. An example would be the stratified squamous found in the skin. And pseudostratified tissues contain a single layer of cells that appear stratified because some of the cells do not extend all the way through the tissue. Pseudostratified columnar cells line respiratory passages and organs of the reproductive system. Two or more tissue types may be organized to form an organ, such as the skin, that performs several specific functions for the body. The skin is a remarkable organ that plays many different roles in our health and survival. It serves as a barrier to harmful organisms. It protects delicate inner tissues from physical or chemical injury. It keeps the body from becoming dehydrated. It plays an important role in temperature regulation by giving off heat. The skin also synthesizes vitamin D in the presence of ultraviolet radiation from the sun and protects us from these harmful rays. The skin consists of two layers, the epidermis and the dermis. The epidermis is composed of 5 layers or strata. The deepest is called the stratum basale. Keratinocytes, the most prevalent cell type, arise there and are gradually pushed towards the skin surface.
Keratinocytes produce a fibrous protein called keratin that gives the epidermis its protective qualities. During their migration to the surface, these cells flatten and fill with keratin and die. When they reach the surface, they form the waterproof layer that allows skin to keep the moisture in our bodies from evaporating.
The stratum corneum, the outermost skin layer, consists of 20 to 30 rows of flat dead cells completely filled with keratin. These cells are constantly sloughing off and being replaced with new keratinocytes from the strata below. Melanocytes, another type of epidermal cell, are found in the second epidermal layer called the stratum spinosum.
Melanocytes produce a pigment called melanin. Melanin influences skin color and also absorbs ultraviolet radiation. The more melanin, the darker the skin. The more effective your protection against harmful ultraviolet radiation.
The dermis lies below the epidermis. It contains the nerves, sensory receptors, most blood vessels, hair follicles, oil glands, and sweat glands. Hair, oil glands, nails, and sweat glands are part of the integumentary system. Each has a unique role in helping maintain homeostasis. The outer layer of the dermis has ridges that reach up into the epidermis, causing folds in the skin surface that increase friction and the ability to grip. These folds are unique for each person and are the basis for fingerprints. The reticular, or lower layer of the dermis, is composed of dense connective tissue, which contains a combination of collagen and elastic fibers. This gives the skin its ability to stretch and return to its original shape. With age, skin loses its resilience. This produces wrinkles and sagging skin. Cells are the basic building blocks of our bodies. From individual, specialized cells to their aggregation into tissues and organs like the skin, all work together in a complex interdependency to keep us alive and healthy.